Hello everyone and welcome to chapter 8. So happy to have you for this lecture. Go ahead and tuck in and start taking notes. Here are our learning objectives for this chapter. Again, I feel like you guys can probably read through this yourself. So we are going to start with telomeres. We've been talking about DNA for the last chapter and a bit, and we need to talk about how cells are able to divide so many times, but the fact that they are actually not immortal. They cannot keep dividing over and over and over. So what happens is we have these little telomeres, which are going to be sort of sections of non-coding repetitive DNA that kind of serve as a protective end to our DNA strands or our chromosomes. Okay? So what's going to happen is that every time we have the replication of these chromosomes for each cell, these telomeres are going to get shorter and shorter. They're kind of like a car's odometer in that they kind of keep track of the mileage of the cell. And in this case, the mileage is how many times the cell divides. And so once we get to the end of the telomere, so we've basically cut off from cell division over and over so many times that the telomere is now too short, that is when the cell itself will die. Because remember, in order to have a new cell, it has to come from a pre-existing cell, but that pre-existing cell then has to essentially, reach the end of its little cell life after it goes through cell division so many times. Telomere length is going to change as you go from an infant to being older. Your body is going to need to experience more cell division as you are growing as a child, and that is going to be, just to give you an idea, your telomeres when you're an infant are able to have about 80 to 90 cell divisions occur before they experience cell death. Meanwhile, when you're about 70, you'll only have telomeres long enough for about 20 to 30 cell divisions. So things really start to slow down as you get older. So when there are malfunctions in the telomeres, you can sometimes get a condition called Hutchinson-Gilford progeria. These siblings shown in this picture both have progeria. This is a syndrome that causes shorter than normal telomeres, so they're particularly short. It's almost as if instead of as an infant, you have those really long telomeres, you have 70-year-old telomeres, which causes the individual to appear to age at a very rapid rate. The average life expectancy is about 13 years for someone with progeria, but they can live up to 20. Some cells are able to rebuild their telomeres. Examples of that are single-celled eukaryotes, like yeast, and cells that produce gametes. So those are those sex cells. And then telomere rebuilding at an uncontrolled rate of cell division, that is what we call cancer. And we'll talk more about cancer later in this lecture. So some chromosomes are going to be circular and others are linear. We've talked about this before, but the big thing I want you to get out of this slide, one, the difference between prokaryotic and eukaryotic cells as far as their chromosomes go, and that is that prokaryotic cells are going to have a singular circular chromosome attached to the cell membrane. Meanwhile, with eukaryotic cells, you're going to have the chromosome, specifically that DNA strand, which we're going to call chromatin, wrapping around proteins called histones. And they are going to form linear chromosomes. So not circular, but linear. So like a line. We've talked a little bit about cell division, just briefly, but let's talk about sort of the mechanics of it. So here we have a prokaryotic cell going through cell division, which is known as binary fission. First, we want to look at this word replication, which is where the cell creates a duplicate of each chromosome. So this is where the parent cell, so a single cell, is going to divide to create two daughter cells. When it comes to prokaryotic cells, this cell division is going to produce two identical daughter cells from one parent cell. And what this is called is asexual reproduction. We're going to talk about the difference between sexual reproduction and asexual reproduction here in the next few slides. But just know this is binary fission. It's only going to happen with prokaryotic cell division. This is that process, but it's actually kind of similar to eukaryotic cell division, but we'll get to that, I promise. As I promised, here we go. So this is eukaryotic cell division, but it's also sort of the whole cycle of the eukaryotic cell cycle. So the first thing I want to point out are the gap phases in this whole cell cycle. So that is that first phase that we see there, gap 1. This is going to be, and I like to think of it as like the infant phase, if you will. It is a phase that is just normal cellular functions taking place for this cell. It's sort of like its adolescent phase in a way. Then we're going to have that second phase, skip over that one that obviously I skipped over. We have S phase. This is very important to make a note of because it stands for synthesis, specifically DNA synthesis. And when you think of DNA synthesis, I want you to immediately think of replication of the DNA. So your DNA is replicating in order to form something called a sister chromatid, and we'll talk about those here in a minute. But essentially it's just making an exact copy of all of the chromosomes. So there's still the same number of chromosomes that we started out with in gap 1, but everything is being replicated. Sorry, my cat is deciding that he's really excited about DNA replication. Next, we're going to move on to gap 2. Gap 2 is going to be this second growth period in preparation for cell division. Now, everything that I have talked to up to this point is going to be all part of interphase. And interphase is something that takes up the priority of a cell's life. So we are going to be in interphase almost all of the time, except when we're in the mitotic phase or going through mitosis. Mitosis is going to be how we get from one cell to two cells when it comes to eukaryotic cells. And then the last little phase is going to be cytokinesis, but I sort of have maybe a different idea than maybe some people. Cytokinesis can kind of be this intermediate phase between interphase and mitosis, but we'll talk about that here in just a minute. So as we are going through all of these different phases, G1, S phase, G2, and mitosis, we need to have a way to be able to sort of check and make sure that everything's going the way we want it to as far as our cells and our DNA. So there are 3 primary checkpoints that will regulate the cell cycle in eukaryotes. The first is the G1 or S checkpoint. This is going to check and make sure that the DNA isn't damaged and is going to check that the cell has sufficient nutrients. The G2 or M checkpoint is going to make sure that the DNA has replicated properly. He is really enjoying these blinds. I apologize for that noise. The last phase is the spindle assembly checkpoint, and this is going to check for spindle fibers being built and attached properly in the points that it needs to be attached. And we'll talk about that when we talk about mitosis. So kind of put a little check in your mind that this is going to be talked about later, and we're going to start talking about mitosis here in just a minute. So here is the process of making a copy of our chromosome, making an identical copy of our DNA. So what this is called is DNA complementarity. And essentially what I want you to know, and I'd maybe write this down if you're taking notes, is that the bases on one strand will always have the same pairing partner on the other strand. And this is a very important concept to kind of understand, because as our DNA essentially splits apart or gets sort of unzipped, we are going to fill in that side, and that is going to result into identical daughter DNA molecules. So how DNA replication occurs is we're going to have an unwinding and separating of that coiled up helix that we were talking about in, I think, the last lecture. So what happens is we have this molecule that's essentially going to unwind our ladder. And then what's going to happen is we're going to have reconstruction and elongation. And this is going to happen with our DNA polymerases. You'll remember that we have talked about RNA polymerase. Do make sure that you know that this is DNA polymerase. So our DNA polymerase, as it's moving through and unwinding and replicating and doing its whole process, it's also going to proofread our DNA and correct any errors that it comes across. And it does a pretty darn good job of it, actually. Most errors are going to be caught and fixed, but if they aren't, mutations can occur, which they happen actually pretty frequently, unfortunately, but sometimes it's not a bad thing. And when it's not a bad thing, that is where we have evolution. So most cells are not immortal, as you can kind of imagine, due to mitosis generating replacements. A lot of the cells in your body essentially move from a baseline and then push the cells above them up and off. So eventually those cells will get sloughed off, which is essentially what dust is, except that's like the sloughed off skin cells. The fastest growing cells on a plant are going to be the root tips, which makes sense. So that's where a lot of mitosis is going to be occurring. You also have a lot of mitosis occurring at your hair roots. So that is pushing the hair up, and so it's growing off of where, you know, it's coming off of your skin, essentially. Sort of like I always think of like a push pop. How you push like a popsicle through the little tube that it's in, and you're eating off the top of it, right? But you're still pushing from the bottom up and out. So this picture here is a skin slide picture, and those layers are going to push up until you see those kind of stringy parts at the top flake off or come off, and that is what dust is a bunch of skin cells. So mitosis has two purposes, that is growth, which we've talked about, and replacement, which we've also talked about. So mitosis is going to replace worn out old cells. So nearly all somatic cells, which you can make a little note, somatic means body cells. These are all the cells throughout your body. And all of these cells are going to go through mitosis, except for your gametes. Your gametes we'll talk about later, but that is a process called meiosis. So in animals, the rate of mitosis varies for different types of cells. For red blood cells, these are all going to be replaced every 2 to 4 months, while the cells lining your intestines are replaced every 3 weeks. So it sort of depends on how much use the cells are getting, how much use and abuse, if you will, if they need to be replaced quicker, if they can last a little bit longer. So kind of fun stuff. This term coming up, apoptosis, is cell suicide. What this means is that cells that accumulate a significant amount of genetic damage, which could potentially lead to cancer, they are known as at-risk cells, are going to be planned to go through cell death. So they might not be at the exact end of their life as a cell, but we are going to essentially kill them off anyway because we don't want problems to arise in their cell division. Cells that go through this are cells like the digestive tract and your liver. Your skin cells are also, in a way, kind of a part of this as well. Moving on to mitosis. So we do have this overview of mitosis. I do want to make a little note. Interphase is not one of the phases of mitosis. Mitosis is composed of a few phases, actually 4, kind of 5 if you count cytokinesis, but interphase is not one of them. So, before we get into all of the steps that make up mitosis, we want to talk about a few things that need to happen before we begin mitosis. And the first being that our chromosomes are replicated. Remember, they have to replicate during that S phase, during interphase. What's going to happen is we are going to produce sister chromatids. Your sister chromatids, and if you look over at the illustration, it is the part that is right underneath the word replication. Our sister chromatids are twin sisters that are conjoined at the centromere, so they are stuck together. And once they condense, then we're ready for mitosis. So initially they're kind of loosey-goosey, as chromosomes have a tendency to be, and then they're going to condense, and that allows us to begin mitosis. And as mitosis begins, we're going to have spindle fibers form, and we'll talk about that here in just a second. But the spindle fibers are going to attach to those centromeres, so that little central hip that our sister chromatids are conjoined at. So very important thing to make a little note of, but we are going to talk about why that happens and what happens after we have those spindle fibers attach. So again, interphase, we have that first part that is not technically part of mitosis. Interphase is going to be that normal time period of growth and development for a cell. We're also going to have interphase be that very important part where chromosomes replicate, and then we enter into mitosis. The first phase is called prophase. Prophase is where we have the nuclear membrane surrounding our DNA, which is now our condensed sister chromatids, breaking down. From there, the sister chromatids will have already condensed or are condensing at the same time. Sometimes it's a little before, sometimes it's during, but regardless, they have to condense in order for mitosis to occur. And then we have our spindle fibers forming, and our spindle fibers are forming from an organelle called the centriole. We've talked about this organelle before, but there he is. There's his purpose, is to form spindle fibers. From prophase, we're going to enter into a phase called metaphase. This is where our sister chromatids are going to line up at the center of the cell. So they create this perfect little line going across an imaginary equator, which is really cool. And how they do this is those spindle fibers attach at the centromeres of our sister chromatids, that's little central hip. And then they're going to start this little game of like tug of war, so pulling back and forth until the sister chromatids line up in the middle. This happens because those centrioles, which have our spindle fibers coming off of them, migrate to the opposite ends of the cell. And you can see that in this illustration really, really nicely. We have the centrioles on opposite ends of the cell. They are attached at those centromeres in the middle of our sister chromatids, and they're pulling back and forth until they line up, and that is metaphase. From metaphase, we enter into anaphase, and this is where those spindle fibers start pulling our sister chromatids apart. So one half of a sister goes to one side of the cell while the other half goes to the other side. Remember, our sister chromatids are identical. So we have one half of that identical DNA going to one side and one half of the identical DNA going to the other. So the same thing is going to either side of the cell. From anaphase, we're going to start to see a little bit of cleavage being created between where those new cells are starting to develop, and that is telophase. Telophase, the chromosomes are going to begin to uncoil, and that is because the nuclear membrane is starting to form, and then cytokinesis is going to start, which again, it's sort of part of mitosis, part of interphase. It's sort of, toeing the line between the two. And cytokinesis is just the cytoplasm being divided into the two daughter cells. So remember, the cytoplasm is all that gooey junk within the cell, including the organelles. So we have two daughter cells. Make a little note. These daughter cells are completely identical to the parent cell. The parent cell has not disappeared. It is now just in the presence of two cells. It is now formed into two cells. So from one cell we have two, and that is mitosis. So in a way, that is asexual reproduction, but not really, because you're not forming a whole organism with asexual reproduction. It's sort of a process of forming a whole organism, like we did with the prokaryotic cells and binary fission. That's asexual reproduction. It's very similar though. So you can see how evolution potentially has, created multicellular organisms by using that process of binary fission or asexual reproduction to form cell division in eukaryotic cells. It's pretty neat. So asexual reproduction occurs when a single parent produces identical offspring, and that means not just that the offspring are identical to each other, they're also identical to the parent. Kind of like cloning in a way. Sexual reproduction is going to need at least two parents. I don't know of any circumstance where it needs more, but at the very least we need two. And this is going to produce an offspring that is unique from the parents and also from its siblings. We are going to need the fusion of two reproductive cells or gametes from the parents, and they're going to need to come together for a process called fertilization. So this is where a sperm meets an egg and you create a zygote, and then that zygote becomes an embryo, and then that embryo becomes a fetus, and then that fetus becomes a baby. And that is that whole process. So that is sexual reproduction. Some species, like in some plants, can actually use both methods of reproduction, which is really cool and very evolutionarily advanced in a way. So sexual reproduction and meiosis. Meiosis is going to be the other process that we talk about to form a different kind of cell. So we talked about mitosis, and mitosis occurs in somatic cells. Again, those body cells. Meiosis is going to be the process to create gametes. Gametes are going to be like an egg cell for a female or a sperm cell for a male. Another really important thing to make a note of is these gametes are going to be something called haploid. Haploid means that it's going to have half the number of chromosomes of the parent. So a diploid cell, every other cell, as well as kind of diploid creatures, are going to have a set of chromosomes. For humans, we have 46 total chromosomes. We get 23 from our mom and 23 from our dad to give us 23 pairs or 46 total chromosomes. That is diploid, having the whole set, okay? Half of that is haploid, and that is what we get with egg and sperm cells. So that when the egg and the sperm cell come together, they create a diploid cell. So once the egg is fertilized, 23 chromosomes from the egg, 23 chromosomes from the sperm come together, fertilize, create that zygote, which is now diploid and has 46 chromosomes. Could I beat that dead horse anymore? Okay, so all offspring have a tendency to resemble their parents, yet no offspring should look identical to their parents or to their siblings. We do have this example picture here of the Kennedys, which they all look very similar. They all had very distinct teeth, as well as hair color and sort of face shape, kind of squarish in a nice sort of handsome way. But the Kennedys all look similar, the Kennedy boys did. And that is because their parents gave all of the Kennedy boys quite a few similar chromosomes in the form of those gametes, but they're not all the same. So meiosis is going to have two important outcomes. It's going to reduce the amount of genetic material in gametes so that, you don't get like 92 chromosomes instead of, 46. And it's also going to produce gametes that differ from one another with respect to the combinations of alleles that they carry. In general, what that means is you're going to have genetic diversity, which is incredibly important for eukaryotes. We want that genetic variation, especially for organisms that go through sexual reproduction. Genetic variation is one of the biggest advantages to sexual reproduction. Okay, so here we are going to talk about something called a homologue, or homologous pairs. Homologous pairs are the same chromosome, but one's from the mom, one's from the dad. So they're a pair that goes together, for like chromosome 1. You've got chromosome 1 from mom and chromosome 1 from dad. Together, they are important as a set because they are homologous pairs. Do make a note that meiosis only takes place in the gonads. For females, this is going to be in the ovaries. For males, it is the testes. And we've talked about how many chromosomes we want to start with and what we want to finish with. So meiosis is going to reduce the genome by half, and I've kind of talked about this before when I was kind of beating that dead horse, but essentially it's this idea that we are going to take through the process of meiosis to create haploid cells or haploid gametes, which are also daughter cells. Don't let that confuse you. We're not saying that all of these cells are going to be female. That's just what we call the end product from one parent cell to haploid or to the offspring of that parent cell. They are known as daughter cells. So what happens with meiosis is we have a diploid parent cell that has our homologous pairs, one from mom, one from dad. And keep in mind that this means that it is not chromosomes from the participating parents creating the new offspring. This is the grandparents, if you will. So if we have, Mr. and Mrs. Smith, this is how they are going to produce the sex cells, an egg and a sperm, to create their offspring. So let's say we're looking at this diploid parent cell that is from Mr. Smith. Mr. Smith has a chromosome set from his parents, so his mom and his dad, they are going to form those chromosomes that he has in order to pass on to his offspring. So as Mr. Smith's diploid parent cell is going to go through meiosis, that information is going to be split up and then split up again. So we go through meiosis technically twice in order to get haploid, which, you know, half of that chromosome information. I know it's a little confusing, but just bear with me. So we've got meiosis. This is division 1. So this is where our homologs are going to separate. These are stages 1 through 3. So again, interphase is, you know, interphase, where our chromosomes are going to replicate in preparation for meiosis. That is the exact same as that mitosis. And then we're going to enter into prophase. And everything's relatively the same, except that our homologous pairs of sister chromatids are going to come together and then cross over. So that is the big difference. From there, we're going to enter into metaphase, and that is where our homologs move toward the center of the cell. At this point, they're referred to as the metaphase plate, and they line up. This is also different from mitosis, and that's what those little red squares mean. Okay. From there, we have anaphase, and that is where our homologs are going to separate and are pulled to opposite poles. Our sister chromatids are going to each side and are going to be a mix of maternal and paternal genetic material. So we have, in a way, sort of jumbled up all this genetic information in order to get some genetic variety and diversity. So from here, we've got meiosis 2 going to be happening. So we have that telophase and cytokinesis occurring to form our daughter cell 1 and daughter cell 2, but we want to go further than that. So we're going to enter into prophase 2, where we have our chromosomes and our daughter cells condense and spindle fibers form. This is the same as with mitosis. So these cells, after we go through meiosis 1, it's pretty much the exact same process as mitosis. So we go through prophase, metaphase, anaphase, and we end with telophase and cytokinesis. But instead of ending up with 2 identical daughter cells, we have 4 unique haploid daughter cells. And that is the big sort of takeaway here, is what do we end up with? Mitosis, the end result is 2 identical diploid daughter cells. For meiosis, it's going to be 4 haploid unique daughter cells. Very important. Okay, so moving on from those processes, let's talk about if cell division gets out of control. What that can result in is cancer, and that's not a good thing. So cancer is characterized by unrestrained cell growth. It can cause serious health issues. It's the second leading cause of death in the United States. The first is heart disease. It is responsible for 20% of all deaths worldwide, and it occurs when disruption of the DNA interferes with a cell's ability to regulate cell division. So this is kind of an overall what is cancer, but let's talk about how cancer cells differ from normal cells. There are 3 primary ways. The first is that cancer cells have no contact inhibition. So while normal cells divide until they bump up against other cells or tissues, at which point they stop dividing because, you know, boundaries, cancer cells don't have those boundaries. They don't know personal space limits. So cancer cells will ignore the signal and continue to divide and pile up on top of one another, which they just keep going and keep going. They're sort of like the Energizer Bunny. The second is that cancer cells will divide indefinitely. While normal cells will reach a certain limit and stop dividing and go through cell death, cancer cells do not have that limit. They will divide and divide and divide for infinity if they can. And then last, cancer cells have reduced stickiness. So while normal cells like to stick together, they're buddies, they would prefer that. Cancer cells are able to break off from each other, and this can lead to something called metastasizing. So you've probably heard of metastasizing before. We're going to talk about it a little bit more in respect to tumors. So we have something called benign tumors, which can be removed safely. That's fantastic. You want a benign tumor if you get a tumor at all. Malignant tumors are not good. These can be inoperable. They also can break off little pieces of themselves and spread throughout the body, and that is called metastasizing, which is not a good thing. The way that they metastasize and move throughout the body is through the circulatory and lymphatic system. So your circulatory is like your blood, and the lymphatic system is sort of like a fluid pickup system. It sort of follows along the same way as the circulatory system. It removes excess fluid from your tissues, but it's throughout your whole body. It also has a lot to do with your immune system, but regardless, however those malignant tumors move around, it's not a good thing. And to answer the question of how cancer kills an organism is that it interferes with other structures and cells. So those cells are going to grow in areas. You don't want it to grow in any area, but let's say you have a tumor or cancer developing in your lungs. That tumor is going to inhibit your lung tissue and the cells in your lungs from performing their normal functions, which obviously is not a good thing, and that will interact with your breathing abilities. So the cancer itself doesn't kill you. Cancer interfering with your other organ systems will kill you. So treatments for cancer, unfortunately, can be as painful and as difficult as the disease itself. If you've ever had a loved one or someone you know go through cancer treatments, you know that it's difficult. So you have two main types of treatment, chemotherapy and radiation. The difference between chemotherapy is that it's a whole body treatment. Essentially, you're pumping a poison into the body and hoping that essentially the poison will kill the cancer before it kills the person or the organism having the cancer. And then the next is radiation. This is much more targeted. It does not go through the whole body. This is going to target one area. An example I can think of immediately would be like prostate cancer. If it hasn't metastasized, it's staying in the prostate. Radiation therapy is really awesome. They'll use like little beads essentially to surround the prostate and target that cancer specifically there so that you don't have to go through chemotherapy. Both of these therapies are aiming to disrupt the cell division process, so mitosis, of the cancer cells. What this does, though, is that it can also affect other cells that also go through mitosis rapidly. This can be skin cells, as well as cells in the intestines, and your hair cells. So that is why your hair falls out, because that process of mitosis is being disrupted. We're trying to attack just the cancer, of course, but what ends up happening is you disrupt the mitosis process, or that cell division process, of the normal cells. So unfortunate things that happen as a way to try to get rid of that cancer. I always think of the way my grandmother has sort of talked about it, and she has used the phrase, it's like killing a fly with a hammer. You know, it's really, really aggressive, and hopefully it works, but you can do some damage to other things by doing that process. There is no cure for cancer, sadly, but technology is moving a long way. There have been very successful stories of chemotherapy and radiation putting cancer into remission. The cancer never truly goes away, but it does go into remission, and that is a fantastic diagnosis to receive. So, moving on from that very heavy topic to how male and female gametes are produced in a slightly different way. So what we have here is an image of the gamete development in female animals, and I know I said meiosis ends in 4 cells, but for females, technically it only ends in 1 viable cell. The other 3 cells are known as polar bodies, which essentially means that they're useless, they're trash, your body reabsorbs them, we don't need them. So for males, the process of meiosis to create sperm cells, which is called spermatogenesis, is going to result in 4 mature sperm cells. For females, this process of meiosis to create one of these egg cells, which is called oogenesis, is going to produce 1 ovum and 3 polar bodies, which are essentially just trash. Okay, so how do we get our genetic diversity through these processes? And that is crossing over. And we kind of glossed over crossing over when we were talking about meiosis, but we did mention it. So crossing over, or genetic recombination, occurs when homologous chromosomes swap genetic information. So this pair, so chromosome 1 from, maternal copy and chromosome 2 from a paternal copy, they're going to just swap a little bit of information in order to spread that diversity. So each of the 4 chromatids are going to be packed into a haploid gamete in the end, but they're not all going to be the same. They're going to have some similar information to pass on, and that's why siblings look similar, or you might look similar to your mom or your dad, but this is how we get that genetic diversity. And where that genetic information being exchanged occurs is called the chiasmata. Little less important information. Just know what crossing over is. So we talked about this ever so slightly. What are the costs and benefits of sexual reproduction? A big benefit to sexual reproduction is genetic differences. So that genetic variation, as I keep harping on, big important thing is genetic variation. A cost, though, is that sexual reproduction is very slow. It's very costly when it comes to energy. And another big one is vulnerability. Vulnerability is a big one. So while animals are mating, they are very vulnerable. While a mother is pregnant, she is vulnerable. And then while she is also giving birth or also nursing the young, she is still very vulnerable. So contrary to that, asexual reproduction, the benefit is that it's very fast. For example, some bacteria can make a new generation in 20 minutes, which is insane and also super cool. But a cost is there's no diversity. So if something happens where this particular organism that reproduces asexually is quite susceptible to maybe temperature, as soon as that temperature reaches a certain point, it's like, boom, everybody's gone. So there's no diversity to make it to where some of the individuals will have a little bit more temperature tolerance than others. So not everybody in the population dies. So to say one method is better than the other, there's costs to both, but there's also benefits to both. So, hey, 6 to 1, half dozen the other. You can decide which one you think is better. So let's test some of your knowledge. I am going to give you about 5 seconds, so if you want to pause on that slide, you can also check your information with the slides that I posted for you. Okay, so hopefully you got some of those answers right. If you have a question as to why you got something wrong, feel free to email me. From here, we're going to move on to how sex is determined in humans, as well as other species. It's kind of interesting, cool stuff. So human sex chromosomes are going to be the X and Y chromosome. So everybody's got at least 2, maybe, potentially, hopefully. Males are going to have the combination of XY, and females are going to have XX. So sex is determined in humans based on the sperm cell that the father produces. But what happens, and something that's kind of interesting, is that females lack a Y chromosome, which tells us that the Y chromosome is not necessary for development of a functioning human. The X chromosome, however, is, because everybody has an X. If you don't have an X chromosome, you probably won't make it to development, but we know a lot of information as far as the X chromosome being very important. Unfortunately, and no offense, gentlemen, the Y chromosome is much less important. It's also substantially smaller than the X chromosome. There's less information that is included on it. So, determining sex in other species, you can have hermaphrodites, which produce both male and female gametes. Examples of these are earthworms, garden snails, and most plants. In humans, we do not call them hermaphrodites. This is something known as intersex. And typically, you've probably heard of this term hermaphrodite. It is essentially a term that is referring to the genitals of an individual, and saying that they have both sets of genitals or parts of both sets of genitals, and that is known as intersex. For birds, the mother is going to determine the sex of the offspring. She can do this as she is producing the eggs. It's kind of cool. Ants, bees, and wasps are going to be determined by the number of chromosome sets an individual has. For example, male ants, bees, and wasps are going to be haploid. That means they have half the number of chromosomes as the females, which are going to be diploid, which, crazy stuff. It's so interesting and so cool. What happens is you'll have a male if the egg is fertilized, or if an egg is unfertilized for a female. It's just so cool. I mean, tell me it's not. From there, something kind of interesting when it comes to turtles, and you've probably seen the way sea turtles lay their eggs in sand. They dig a nice big hole, and then they plop their little clutch of eggs in there, and the environment is going to determine the sex of the offspring, specifically temperature, and it comes down to like literally like a degree will determine whether or not the egg is going to produce a male or a female turtle. So kind of cool. Okay, so over to the right here, I do want to point out that this is something called a karyotype. This is where we can see every chromosome of an individual, and of course you can see that they're in pairs, one from mom, one from dad, for each chromosome. And then at the end we have our sex chromosomes. So there are 23 pairs in total, or 46 altogether, and then that last one doesn't have a number. That is either going to be XX or XY. And in this case, we are going to start talking about what kind of happens with birth defects in respect to chromosomes. So there was a study that looked at Down syndrome and mother's age of the offspring. So what they found is as the age of the mother increased, so did the incidence of Down syndrome. So what that tells us, and honestly you could do this with just about any genetic condition where a mother is older and she is having offspring at an older age. The same goes for fathers. If the gametes are older or being produced by an older person, there is a higher likelihood of passing on genetic information that is going to lead to maybe unfavorable conditions for the offspring. So there are two different methods that can be used to collect cells from a fetus in order to sort of screen for any conditions that may be developing that maybe the sonograms don't show. And I am going to struggle with these words. Amniocentesis, there we are, is a sample that is taken from the amniotic fluid surrounding the fetus inside of the mother. So she has the placenta, which is sort of like an eggshell surrounding the fetus that is developing, and the fetus is floating around in something called amniotic fluid. It's sort of like a baby broth, if you will. It's a nice fluid that's going to act as a cushion and also nutrients for the baby. The second is chorionic villus sampling, or CVS. These are cells that are obtained from the placenta, so that outer covering for the baby. So trisomy 21, that is the technical terminology for Down syndrome. It was first described by Dr. John Langdon Down in 1866. It was revealed by the presence of an extra copy of chromosome number 21. So that means that there are 3 copies of chromosome 21, and this will affect 1 in 1,000 children. Going sort of hand in hand with trisomy 21, that is an example of something called non-disjunction, and that is an unequal distribution of chromosomes during cell division. Cases of trisomy tend to involve only chromosomes with the fewest genes. These chromosomes can include 13, 15, 18, 21, and 22. Other chromosomes that have non-disjunction will essentially either lead to death or a lack of development of something important like a skull or a brain or a heart or things like that. It's not a good time. Problems associated with reproduction can increase as a woman ages and is having offspring, which we've talked about before. So life is possible with too many or too few sex chromosomes, and here are a few examples. So this first one is called Turner syndrome. This is going to result in a female who only has a single X chromosome, which is really, really cool. So what happens is this individual will have a shorter height, a web of skin between the neck and the shoulders, underdeveloped ovaries. They're often sterile and can experience some learning difficulties. So they only have one sex chromosome. Meanwhile, with Klinefelter syndrome, you will result in a male who has underdeveloped testes, a lower testosterone level. They're usually infertile, and the development of some female features may occur. This is going to result in an individual who has a tendency who is male, but who looks a little androgynous or a little bit feminine. So this individual has 2 X chromosomes and 1 Y. And you can see on the left where they're getting it from. So the mother's egg has 2 X chromosomes, and then the male father is going to produce 1 Y chromosome, or it's going to be the mother has 1 X chromosome and the sperm cell has an X and a Y. So kind of interesting stuff, I think. The last 2 examples I have involving sex chromosomes are XYY males, which can sometimes be called super males because they have 2 Y chromosomes. They're normally taller than average, and moderate to severe acne is also a condition. Their intelligence may be slightly lower than average, and oddly enough, and the study's really biased unfortunately, but quite a few XYY males seem to have like I said, potentially higher testosterone, but end up maybe having less ability to perform good judgment. So quite a few XYY males are actually incarcerated. So it may be a genetic thing as to why that happens. So the last example is an XXX female. They're usually sterile. There's no obvious or physical mental problems, but these can usually or sometimes be called meta-females. So kind of neat. There is one last question. Hopefully you get it right. And that is the end of chapter 8. As always, make good choices, have a wonderful day, and if you have any questions, do not even hesitate to send me an email.